Okay, awesome. Thanks, James. So today I'm going to be talking you guys through one of the it's a very important topic in physiology, but it's going to be explored a little bit more in pharmacology. So they've kind of split it up in a bit of a strange way, where in physiology, you'll just be covering sort of the basics of the different systems within the autonomic nervous system. And then in pharmacology, you'll be exploring it in a bit more detail in terms of the subtypes of all the receptors and things. So I'm not going to go into all of that detail this week. I believe that will be scheduled for next week. But hopefully today will give you a bit of a platform to be able to really understand the effects of each of the systems, so the physiological effects. And I believe that you may have covered this at a very preliminary level last semester. So you may notice that a lot of this does repeat a tad bit, but I'll be going into it in a little bit more detail with you guys. And let me know if there's anything that you have questions with, because I did put these slides up based on basically your lecture slides and some of the notes that I made. If you guys do find that there is some extra things that your lecturer may have talked about this year, let me know and I can just explore those as well. But I don't think that it would change that much. Also, how good is it that there's no anatomy this week? That's awesome. So I'm going to be starting off just with a general overview of the peripheral nervous system. So you'll notice that I put a lot of ticks everywhere. That's just some of the stuff that we've already discussed so far. So we've discussed a little bit. I mean, we haven't really talked too much about the brain and the spinal cord. We have a little bit, so you may have a general idea of how they're structured. And peripheral nervous system, we've talked about what it is. So we know that it consists of sort of your sensory division and your motor division. Sensory division, we had a really great introduction to, I think, with James, talking about all the different sort of brain regions that are responsible for sensation and all the different receptors in your fingers and things like that. So we already discussed that in quite a bit of detail. Today, we're going to be exploring the motor division a bit more. But the interesting thing is that you'd think the motor division is all sort of skeletal muscle. The only thing is that there are actually some motor nerves that go towards innervating organs within your body that have sort of their own intrinsic movement. And so it's not necessarily to do with just your voluntary skeletal muscle. It can be all those organs as well. And if you break that up, so this is the autonomic nervous system that we're talking about. If you break that up, you'll notice that it has sort of two other divisions, sympathetic and parasympathetic. So we'll be exploring each of them in a bit more detail in the next couple of slides. Okay, so the peripheral nervous system generally, or this is more so talking about the autonomic nervous system, its main function is to maintain homeostasis and help modulate intrinsic organ activity. And I'll show you what that means when we go into the effects of each system. But just remember that overall, this is all trying to make sure that everything within your body is running within a set degree of parameters. So you don't want your pH to be way too high or way too low. You don't want your carbon dioxide levels to be too high or too low. You want everything to be just right. So that's what this system is talking about, is trying to get them within those narrow barriers that we consider to be safe. Now, just a brief comparison between the somatic and the autonomic. If you have a look on the left-hand side, somatic, you'll see, is generally just a very simple system. So you've got your motor cortex, and we went into all this detail previously. Got your motor cortex, it's going to send off a nerve that goes down into the spinal cord, and then synapse at generally the level of the muscle fiber. And then that motor fiber will go and innervate the muscle fibers and then cause contraction or relaxation. Now, with the autonomic nervous system, it's a bit more complicated. So I kind of want you to ignore the section that is prior to the spinal cord. We're going to focus on spinal cord and then going out. So with the somatic system, you'll notice it's just one nerve going out. With the autonomic nervous system, it's a bit more complicated, because once everything leaves the spinal cord, you've got one nerve coming out, and then you've got it synapsing with another nerve. So we generally call it a two-nerve system. And the length of those nerves can vary depending on which branch you're talking about. Another name for autonomic nervous system is called the visceral motor system. That kind of, I think that's a bit more apt in terms of describing what it does, because you've got the motor part in it, which tells you that it's doing some sort of action. And then visceral is just another word for organ. So it's acting on the organs and causing some sort of movement of those organs or some sort of effect in those organs. It has three divisions in general. We've got sympathetic, parasympathetic. These two words you may be familiar with. Enteric is another system that is generally classified under autonomic, but we won't go into that this year at all. That will be next year when you're talking about gut. But I thought I'd bring it up just in case next year you see that term, you're not unfamiliar with it. And so that's like the autonomic nervous system of the gut. It's kind of the gut brain. Everything else, though, sympathetic and parasympathetic, applies to every single or most organs. So again, just to reiterate what I've been saying, the top diagram is showing you the somatic nervous system. And you can see that from the spinal cord, there is just one motor neuron. And if you remember from our previous studies, it's going to be releasing acetylcholine. And generally the receptor that it acts on is going to be called a nicotinic receptor. And those nicotinic receptors are always going to be, they're always going to be stimulatory. So they'll always cause contraction of the muscle. That's different to the autonomic nervous system, where there's actually a lot of variation depending on which sort of nerves you're talking about. But again, as a broad classification, you will always have two nerve fibers. And in the middle, so where there's synapse, you will have what's called a ganglion. Now, the cool thing is that these ganglion can actually sort of chain together. And I'll go into that in a couple of slides. But just remember that term for a bit later. All it means is that it's a synapse between the first nerve and the second nerve before it reaches your target. Then another really important thing is that it doesn't necessarily have to be just stimulatory or excitatory. It can either be, it can be either of those. And another really interesting thing is that if I say sympathetic, it's not necessary that every single sympathetic nerve is going to be excitatory. And by converse, it's not going to be completely true that every single parasympathetic will be inhibitory. It actually depends on the target organ that you're at as well. So there's a lot of variation happening here, and it's important to understand that. You will go into this more in pharmacology. Also the naming, the nomenclature, the nerve that comes before the ganglion is called preganglionic. That's just the one that starts from the spinal cord. And the one that goes to the target organ, in other words, it's after the ganglion, is going to be called the postganglionic nerve. All right, here's just a summary slide. This will be useful for your revision if you're having any troubles putting all this knowledge together. I know there's a lot of slides in the lecture that have a lot of different things placed everywhere. So hopefully this makes it a bit easier. In terms of the sympathetic and parasympathetic systems and then nerve roots, the reason why I put nerve roots is because the general idea that most of us may have is that when I'm talking about sympathetic, the way that I define a sympathetic nerve is that it causes a flight or fight response somewhere. So it's going to activate a response. It's any nerve that if it were to be activated, it would cause something that would aid me in like running from a bear. So for example, sweating would always be sympathetic. For example, vasodilation in some parts of my body will always be sympathetic. That's actually not the way that these nerves are defined. A sympathetic nerve is actually defined by its location on the spinal cord. So if you think about your 4 divisions of the spinal cord, and I'm not sure if you've gone into this in a heap of detail. So if you want, you can have a diagram of the spinal cord in front of you with like the 4 or 5 divisions. Just remember that for sympathetic, it's going to be the thoracolumbar region. So it's kind of like in the middle 2 divisions of the spinal cord. And specifically, we're talking about T1 to L2/L3. There's a little bit of debate, but generally that's going to be what holds T1 to L2, L3. Parasympathetic then is actually really interesting because if you think about the name, para means on either side of. So parasympathetic means it's on either side of the sympathetic nervous system or the nerves. So sympathetic was in the middle. Parasympathetic will be the one above and the one below. So in other words, it's going to be the cranial sort of nerves and the sacral nerves near the bottom. And specifically, we're talking about cranial nerve 3, 7, 9, 10, S2 to S4. So that's the sacral region at the bottom. So if you remember parasympathetic and why it's called that, that'll make it a bit easier to remember which one is which. The ganglion, so remember that part where the preganglionic and postganglionic nerves innervate each other, or synapse. With the sympathetic nervous system, it's generally going to be closer to the spinal cord. And that kind of leads into the length aspect as well. Because if I was to draw, I might just do a quick annotation. I think I have it on the next slide, but just to give you a bit of context, with the sympathetic nervous system, you will generally have this is from your spinal cord. You will have this preganglionic nerve, and then you will have a nerve going all the way to the target organ. That's generally what your sympathetic will look like. With your parasympathetic, the preganglionic will be longer. And then at the end, this postganglionic will be a bit shorter. That's a very bad diagram. I just had a look over it. The next slide will show you a lot more information, but just to give a bit of context now, that's what it looks like. And the reason why is because generally, the parasympathetic nerves, let's remove all this annotation. The parasympathetic nerves will, like the postganglionic parasympathetic nerves will be much closer within the target organ. So one example might be the heart. And if you go back to a couple of slides, you might go back to the heart and realize that, the preganglionic was quite long coming from the spinal cord. It only synapses with the postganglionic fiber within the heart tissue itself. And so the postganglionic fiber is like this small, and then the preganglionic is this big. Okay, whereas the sympathetic would be the opposite. So that kind of makes sense why the ganglion is going to be closer or further away from the spinal cord based on the lengths of those fibers. Now, in both of them, the preganglionic fiber is always going to be myelinated. It's like remember, myelin is what increases this transmission speed of the signal through the nerve. So it's going to be the faster one. And then postganglionic will be unmyelinated. So it's sort of like in my mind, I just think it's coming straight from the brain. So the preganglionic is going to be the super fast one. And then later on, it might become unmyelinated, a bit slower. I'm not sure what the reason is why the other one is unmyelinated. I don't know why it's to slow it down, but it's just a general rule. The first one will be myelinated, second one unmyelinated. Now, transmission wise, again, a little bit more detail in the coming slides. At the ganglion of both of these systems, so where the presympathetic synapses with this, the preganglionic synapses with the postganglionic, you will always have acetylcholine, and it's actually at an N receptor. So remember that nicotinic receptor. So in other words, at the ganglion, you have the same sort of interaction happening as you would have in the somatic nervous system, which also has nicotinic receptors, acetylcholine. The main important thing, though, that differentiates them is what's happening at the target. So once you've gone all the way to the end of this two nerve system, with the sympathetic, you will have noradrenaline. And that kind of makes sense because if you think about the flight or fight, you're thinking more along the lines of adrenaline. And that'll innervate at the alpha or beta adrenergic receptors, so adrenaline receptors. With the parasympathetic at the target, you will have, again, acetylcholine. So same as at the ganglion, same as at the somatic nervous system, but instead of having an N receptor, you will have an M receptor. The difference, again, pharmacology will do wonders with your knowledge on this. The N receptor, nicotinic, is usually always going to be excitatory. Remember the skeletal muscle. You kind of just want to excite them most of the time when you're activating them. With the M receptors, so muscarinic, those can actually be either, and it depends on further subclasses. So all of these will have subclasses. And then finally, for the effect, this one is probably the easiest one to remember. With the sympathetic, it's going to be flight or fight. So if you're running from something, if you're scared, usually this will be activated. With the parasympathetic, it's going to be rest and digest. So once you're relaxed, what are the things that you'll be doing? And you can start, drafting a list of, if I was to think about sweating, would that be activated during sympathetic response or parasympathetic response? That's probably a bit of a tricky one just because it's an exception to some of the rules, but I'll try and give you an easier one next time. Just think about some of the things, like for example, your airways. Do you want them to constrict when you're trying to run from someone, or do you want them to dilate so that you get more air going through when you're trying to run from a bear? Okay, so think about those things and think about the relation to flight or fight, rest and digest. So here are two diagrams that really helped me or served me well when I was revising, especially the right-hand side one. The left one is a bit more complex, and that's sort of, so substitute my crappy diagram for this one, and you kind of get a very good understanding of what the different nerves are doing. This one really sort of demonstrates the length of the fibers quite well. So you can see with the sympathetic nervous system, ignore the one with the adrenal medulla in it. So that's like a slightly different version, and I'll ignore that for now. I'm not sure if you go into it this semester, but if you do, then let me know and I can explain it a bit more. With the sympathetic, the presynaptic, preganglionic will be shorter, and then the postganglionic is much longer, as you can see. And then with the parasympathetic, it's the opposite. Okay, and you get some sort of idea of what it innervates. So remember, skeletal muscle is somatic. Everything else, like smooth muscle, all the glands, cardiac muscle, gut tissue, all of that stuff will be autonomic derived. The right-hand side is more for revision. So if you're able to draw that out from memory, then you should be really well prepared for the exams. Like if they ask you for the sympathetic nervous system, what is the main neurotransmitter at the target organ, then you specifically know it's going to be noradrenaline. Just a quick note, like in American textbooks, I think they write norepinephrine. So probably don't write that in an exam. I don't know if that's going to lose you marks or anything, but just write noradrenaline. And probably the more important thing is not to write adrenaline. So adrenaline is slightly, like some of them do release a bit of adrenaline, but I believe that noradrenaline is the more potent and of the more common sort of neurotransmitter that's released by sympathetic. For parasympathetic, it'll just be acetylcholine. So as long as you remember that, you'll be very well served for the exams. And the bottom is just a bit of like just telling you for acetylcholine, remember that we're talking about the context of just sympathetic, parasympathetic, but acetylcholine is going to be in both of the motor neurons as well as in the parasympathetic nerve endings. Plus, if you look at the middle, where the ganglion are, there's always going to be acetylcholine, wherever those ganglion are. So acetylcholine is by and far very, very common. The only place that you will get noradrenaline is at the end of the sympathetic nervous system. So that might help you a little bit as well in terms of figuring out which neurotransmitters act where. Here's a good diagram or a good summary slide showing you the effects of the sympathetic and parasympathetic nervous system. I don't think that, you don't need to memorize a whole list, because as I said, just think about what happens when you're panicking. So if you're sitting an exam, what are the sort of effects that are going to be happening around your body? And then what are the effects that will happen once you finish the exam when you're home and just watching TV? That'll be sympathetic and parasympathetic respectively. So for sympathetic, I want my pupils to dilate. In other words, that's called mydriasis. So I can get more light in and I can view things more clearly. Bronchodilation, as I said, so that's going to be dilation of your airways to get more air in. Tachycardia, so you get increased heart rate, and that's going to increase your cardiac output. So you get more blood pumping around to every part of your body so you can actually run. Vasoconstriction is generally the response that you'll get, except for to skeletal muscle. Now, just to clarify this, that's, if you think about it, sort of makes sense, because you want more blood to be going to skeletal muscle when you're trying to run versus to your gut, because you don't want blood to be going to your stomach when you're not trying to digest anything while running. You wouldn't want to be digesting food while you're running, because that sets you up very badly for wanting to go to the toilet while you're running, which is not a good thing. So every other part of your body is going to constrict in terms of the blood vessels, except for skeletal muscle. And you also have decreased GIT motility. So you don't want food passing through too quickly, because then you'll have to go to the toilet. And another one that's not really mentioned here is your bladder. So you don't want your bladder to be emptying while you're running as well. So it'll inhibit emptying, as well as constrict some of the sphincters that allow you to urinate. Parasympathetic is just the opposite of all of this. So instead of dilating, you will have constriction of the pupils and so on. Bronchodilation, bronchoconstriction. Some extra ones that you will see in pharmacology will be emesis. So that's just vomiting. Lacrimation, which is crying. And salivation, which is producing saliva. And so salivation sort of makes sense, because that's a position where you are in the right spot to be able to eat. You're not running away from anything. Emesis and lacrimation are a little bit more difficult to understand. But if you think about sort of what emesis and lacrimation, like they're sort of, you require energy to be able to do these processes. And you don't want that energy to be being used up while you're running from something, right? In the sympathetic nervous system. So in this spot, in the parasympathetic nervous system, these things can be allowed, emesis and lacrimation. And this kind of sets up really well for when you're doing some of the drugs or the medications that are useful in the autonomic nervous system. These can be some of the side effects if you have overactivation of the parasympathetic nervous system. You can get vomiting and crying, okay? So just keep this slide in mind for when you're doing pharmacology. So this is what I was talking about when I was saying pathways from like where the nerves are going. As soon as they exit the spinal cord, they don't just travel in the same direction that all of your other motor neurons do. So I did mention that under the motor neurons, this visceral motor neuron will not have the same path. What it does is that, I'm not sure if you can see my cursor. I don't think you can, but if you just follow the sort of green neuron, remember that's our preganglionic. So it starts off in this spot here. Now this looks like the sort of the ventral horn, but it's actually got a specific name. It's called the IMLCC. I would encourage you to at least remember the name, just in case it has come up in a multiple choice, but I don't think it's ultra high yield. It's just this little intermediate bit between the ventral and dorsal aspects. So intermediolateral cell column. And then you've got, so when you've got that nerve exiting from the ventral horn, it enters the ventral root. Up until this point, you can't differentiate it from just a general motor neuron. However, instead of exiting through this common spinal nerve, it'll sort of divert back and go into this ganglion. Now, this is what I meant by ganglion, because if you think about it, there's going to be the interaction between the first neuron and the second neuron's body. So this will actually cause a massive dilation of nerves. So anytime you see a ganglion, the word ganglion, just think about there being a lot of synapses happening there. And that's why it looks like a big clump of nerves, because there's a lot of those different interactions happening. Now, the neuron will have a choice from here. either you can see the dotted lines. It either shows that it'll stay at that level, or it'll go up and it'll go down. So you may have had a video from Michelle Lazarus potentially talking about this sort of concept of like an elevator, where your sympathetic nerves, at least the preganglionic or the parasympathetic preganglionic, can choose to stay at the level at which the spinal cord is at, or it can go up or down. And if you think about where the sympathetic nerves are, it's in the middle section of the spinal cord, right? Thoracolumbar. If that was the case, you would only be able to get sympathetic innervation at the same level as the thoracic region and the lumbar region, which is not very good, because I may want sympathetic innervation to go up to my brain. I may want sympathetic innervation to go down to my legs. Same with parasympathetic. It's only at the top and the bottom. If I want it to be in the middle, how am I going to get there if it's just going to be at the same level? And the way that happens is through this elevator. So I'd encourage you, if you can, to have a look at this diagram and just research it, remember it, and try and draw it out from memory so you understand specifically where all the synapses are happening. The main key important things to remember are that it's the preganglionic neuron, not the postganglionic, so the green one, that is able to either stay at the level or hitchhike, go up, hitchhike, go down. And only when it decides, yes, I really want to go to the brain and I'm going to stop here, or I really want to go to this vessel in the leg and I'm going to stop here, then it will decide to then innervate with a postganglionic neuron. And then that postganglionic neuron will exit, go the same direction as any motor neuron would, which is via the common sort of spinal nerve. Okay, hopefully that makes sense. If there's any questions, again, let me know. You'll cover this in a lot of detail next year as well. Now, here's just a very brief, very brief overview of some of the receptors that you can get with the autonomic nervous system. So with cholinergic, so remember cholinergic just means acetylcholine. So remember what I said, you have the parasympathetic, right, which has acetylcholine in it. You also have your somatic, which has nicotinic in it. Now within both of these, and also within adrenergic, so adrenaline receptors, which are in the sympathetic, you will have subtypes. For muscarinic, you will have everything from M1 to M5. For nicotinic, you technically have NM and I think NN. And I think you may be familiar with what those mean already. With the NM, it's the neuromuscular junction, so it's at the very end. And I believe NN is sort of in the middle. So where the ganglion are, that's what NN stands for. That's not really that important to remember. The alpha receptor and the beta receptors will also have subtypes. So alpha receptors generally A1 and A2. And really scaringly, there's also subtypes to those. So like there's A1A, or alpha 1A, alpha 2B, and so on. Then with beta receptor, you've got beta 1, beta 2, beta 3, and also subtypes underneath them. Now, don't stress too much. You will cover this in pharmacology, and our team will do a very good job of going over it. So you won't have to worry about it too much. Just know that when you read through this lecture the first time or watch it the first time, it'll be very confusing. But stick through it because it's going to be quite important. There will be a couple of questions in your exam that are to do with this. So keep that in mind. And here's just a summary diagram for you for every single thing we've just talked about, including the receptors at the end and some of the general tissues that they may innervate. Okay, now just a brief talk about the central control of the autonomic nervous system. So the autonomic nervous system is not just running rogue by itself. It actually gets inputs from our central nervous system. So they're not two independent things. They actually collaborate with each other. And there's a couple of good reasons for this, which I'll go into. So keep that in mind. Central nervous system also has an effect on the autonomic nervous system, which will also have an effect on the rest of the organs in your body. Or that meme there, if you don't understand my words and want to put it into pictures, CNS controls ANS, ANS controls organs. Now, the left diagram shows you a bit of a brief sort of big picture overview of all the different components that are acting. So if you look at where the hypothalamus is, that's going to be the bulk of what I'm going to be talking about. The brainstem is also going to be a section that I'll talk about. Limbic, so emotional input, I think that one may be kind of self-explanatory, because if you think about your emotions, and if you think about, for example, when you're in an exam and you're super stressed, you're really scared that, oh, what if I fail? What if I don't do well in this? Which you shouldn't be, because you're really smart. But if you think about it, like, oh, what if I'm going to fail? That's an emotional thing. That's what you're feeling. That's sort of a voluntary thing that you're feeling, right? However, that's going to impact on your autonomic nervous system because you will start sweating. You may go red because of vasodilation to your skin. You get all these different effects that are happening, which you're not technically in control of, but you technically are because of your emotions. So central control does have a direct impact on autonomic nervous system. And how it works is that generally the CNS will receive sensory information. So for example, in that exam, I look at the paper, the first question has something about where are the nerves of the autonomic nervous system starting? And I don't remember that it's the IMLCC, and I'm panicking. So I'm looking at that as my sensory input. What that does is it delivers it to the CNS, and then my CNS will have a control over the visceral motor nerves of the ANS to maintain some sort of homeostasis. So I'm super stressed. It'll let out a little bit of sweat to maybe help me with my stress. Not that that really helps, to be honest. So the primary organs we'll talk about are going to be hypothalamus, brainstem. I think your lecturer goes into a lot of detail, but I've tried to make it as condensed as I can. For the hypothalamus, just remember that it is around about there. It's a very small spot, kind of like underneath the brain a bit. And it is basically the reason you're not dead right now. It is the big boss of the ANS central control. I think the whole brain in general is the reason why you're not dead. But hypothalamus is even more so important because it is the big boss. It controls all your ANS things. So if you didn't have this as part of your body, you may realize that you're not able to control the dilation of your airways. And so you may not be able to adapt to changes in air pressure, or whether you're running or you're walking. And so you will very quickly not be able to get oxygen supply. That's just one example. There's so many examples of why this could be bad. So what it does is that it receives inputs. So if you go back, it receives a lot of inputs, which is why it's called the big boss. You've got from the limbic lobe, you've got from the cerebral cortex all the way at the top. And I know that these arrows are all pointing downwards, but they also have some feedback mechanisms. So the brainstem will communicate backwards. Spinal cord will communicate backwards with the hypothalamus. And the hypothalamus is sort of integrating all this information and figuring out what the hell am I supposed to do with this? How am I supposed to make sure I maintain homeostasis? Interestingly, it can access sensory information from everywhere, including outside the blood-brain barrier, because it has sort of extensions that go out from the, remember the blood-brain barrier, I'm not sure if you've covered this, is sort of the barrier that prevents certain compounds from getting up to your brain. And that can be useful as a survival mechanism, but you want your hypothalamus to act as a bit of a sentinel. If it didn't have that information, it wouldn't be able to tell, for example, how much glucose there is present within the blood, or how much CO2 that's present within the blood, and then make an informed decision based off that. So it needs a bit of secret sort of special access that the rest of the brain doesn't. It can also be, as I said, modified by inputs from the limbic system. So memories and emotions, I've already talked about this. And it maintains homeostasis through kind of comparing whatever you have right now, so pH levels, CO2 levels, comparing it to what should be there, so the biological set points, and trying whatever it can to get it to that level, okay? The way it does this, or the 5 main sort of mechanisms it uses to control homeostasis are listed below. So it controls your blood pressure and your electrolytes. Those sort of go hand in hand. You will cover this next year as well. It regulates body temperature, controls your energy metabolism, regulates your reproduction. So that's super important as well. And it controls your stress response. All of these things are this one really small part that's sort of tucked into the bottom of your brain. Then the brainstem, I'm sure you may have seen potentially the brainstem in TV shows if you watch any medical dramas or stuff, of like a patient coming in with a tumor in the brainstem, or they've got to do a brain surgery and the doctor's like, oh, I can't do it because it's too close to the brainstem. You were thinking like, what's wrong with the brainstem? Like, why is that so important? The reason why it's so important, it's maybe not as important as the big boss, the hypothalamus, but it controls all the flow of messages between the brain and the body. That doesn't really do it justice because it's got 3 parts. It's got the midbrain, the pons, and the medulla. Now, I don't know why, I just remember this. I think in your lecture, the lecturer may have described the brainstem as being like a pregnant alien or something. I'm not sure if that rings any bells for anyone if you've watched the lecture. I don't know how that helps for me personally. I guess it kind of makes sense. You can see the head and you can see the whatever. But if that helps you remember the shape, just to remember, oh, this is actually the brainstem, then go for it. In terms of the contents or the controls, the midbrain controls generally sleep, consciousness, vision, hearing, temperature regulation. The pons, which is the middle bit, or the belly, I think, of the pregnant alien, is going to be controlling the motor control of organs, like the eyes and the face. It's going to control hearing. It's going to control swallowing. I think the medulla is probably quite an important one because it controls most of your vital functions. So it controls things like your general respiration and your GIT motility. So whenever the doctor was saying, we can't operate on the brainstem, because if they damage the brainstem, then you would probably slip into a coma or die, because you would like, for example, you may damage the part that's controlling your breathing. Remember, you don't think about your breathing, it's happening by itself. So if you damage the part of your brain that's controlling all that, you're going to die immediately. You won't be able to breathe at all. So that is, it's really, really important. I think the main takeaway from this central control aspect is that the hypothalamus does all the big muscle work, controls these 5 sort of mechanisms. If you can remember them, then you're very, very well off. And the brainstem is basically something that you cannot live without, I guess. That's basically all I remembered. If you damage it, you die. Okay. So hopefully that helps consolidate some of the stuff you may have watched in your lecture. And if not, if you haven't watched the lecture yet, hopefully this gives you a good way to structure your notes or whatever you're doing to revise. And I think that's it. I'm just going to have a quick look at the chat to make sure I haven't missed any questions. But I feel like you'll have doing a very good job on this.
All right, thanks, John.
Yeah, that looks good. Okay, I'll pass it on to Deepak.
Yep, thanks, Son. So I think moving on to the next part, which is going to be discriminative somatic sensation. So we just talked a lot about the autonomic nervous system, and what we're really referring to when we talk about the discriminative somatic sensation is really different sensations we feel and the receptors that sort of facilitate those different sensations. So starting us off, simple somatic sensations like mechanoreceptors, there are 4 main types, and they detect touch, pressure, and vibration. On the picture on the right, you can sort of see a bunch of stuff in the dermis of the skin. And if you have a look at these different types of cells, so we've got the Merkel cell, Pacinian corpuscle, Meissner corpuscle, and yeah, that's the kind of stuff we're referring to when we talk about sensory receptors that lead to mechanic reception, sort of. So there are 2 main categories of mechanoreceptors. There's slow adapting or fast adapting. Slow adapting is referred to as tonic, fast adapting as phrasic or phrasic. The idea is that slow adapting receptors would create nerve impulses for an extended period of time as long as the stimulus is applied. But fast adapting mechanoreceptors adapt really quickly, which causes them to essentially respond to changes in the environment rather than just a stimulus. So if a stimulus prolongs, it will eventually stop firing, but if the stimulus changes, then it will start firing again. So it basically adapts to its surroundings faster. So we're talking about 2 different types of each of those things. So that's we call that SA1 or SA2. So slow adapting 1 and slow adapting 2, Merkel, Ruffini, Meissner, and Pacinian. If you have a look here, this diagram sort of depicts all of them together. So we've got Merkel, which is an unencapsulated mechanoreceptor of a small receptor field, and it has high density in the fingertips. So if we look at the Merkel's disc or Merkel's cell, you'll notice that it's closer to the surface of the skin on the border of the epidermis. I mean, well, in between the dermis and the epidermis. And that's important to us because that sort of changes the way that we view these different mechanoreceptors. So this is a diagram, I mean, a table, which I think is very useful. It sort of illustrates and categorizes these mechanoreceptors pretty well. So we just talked about Merkel, and you can see this on this part of the, on the first column here. And you can notice that there's slow adapting 1, 2, fast adapting 1 and 2, but slow adapting 1 and fast adapting 1 have a bunch of similarities. So for example, both of them are located in the upper dermis. So as we just saw, the Merkel's cell or the Merkel's disc was on this sort of like at the edge of the dermis, bordering into the epidermis. And both of them have a small receptor field. Both of them have high spatial resolution and density, and exist in non-hairy skin predominantly. Going back to here, we're talking about Meissner's corpuscles. Yep. So you can sort of see like how they're spatially sort of in the same location. And they respond to light touch and low frequency vibration. I brought up the word receptor field a couple of times, so let's talk about that a little bit. So a receptor field is essentially an area where any simulation or any stimulation is discerned as being from one stimulus only. So as you can see on this diagram, these sensory neurons create sort of like a zone that they control or they can detect stuff in. If you have more than one stimuli, if you have two stimuli in that one receptor field, it still ends up creating the sensation of something being in that zone, but you're unable to localize it to two different stimuli. So you'd still sense it as one stimulus, despite the fact that you have two different, technically like, two parts of this is touching the skin, right? When you have overlapping receptor fields, you have better, I guess the word was spatial resolution, because you can detect where things are more accurately, since when they overlap, you sort of create the opportunity for different nerves to also sort of receive information from one side. We also talked about phasic or tonic, maybe not phasic. And like I said, it's related to the rate of adaptation. So as you can see on this diagram, the phasic one rapidly adapts. So if we were to generate a stimulus, it would, the stimulus we're generating can be seen on the third row. So we're just going to apply something, keep it there for a while, and then remove it. When we apply it, we actually get the phasic receptors firing. When we maintain that application of that stimulus, the phasic receptors adapt really quickly and they don't fire. But when it stops, the phasic receptor starts firing again. For the tonic receptors, you can sort of see how it's different. It adapts much more slower. So it starts firing, but then slowly decreases in firing rate. So we've sort of been jumping all around the place. So reorienting ourselves to what we're looking at, we have 4 different types of receptors: slow acting, fast acting. We have similarities between slow acting 1 and fast acting 1. Similarly, we have similarities between slow acting 2 and fast acting 2. So since Ruffini and Pacinian corpuscles are further away from the surface of the skin, they generally have larger receptive fields and are more sparser in number. And for example, Pacinian does. And this leads to like different sensations, because if it's deeper down, we can sort of use that more for pressure and vibration, and wouldn't be as useful for like light touch, right? So going to this slide, as you can see, SA2 and FA2 are similar as well. I like to think of it as MR, MP, Mr. MP. Just a quick way to like sort of remember what letters they start with. It's up to you how you decide to remember it though. But when it comes to the senses that they sort of detect, you can see that, well, yeah, it's listed here. So like in terms of functions and the senses they have. But yeah. Moving on to thermoreception, we just talked about mechanoreception. Thermoreception is the sensation of feeling changes in temperature, so heat or cold. And it's important to understand that these are changes in temperature from a set baseline. Because there's no such thing as an objective warm thing or an objective cold thing. It's just a matter of if you feel it's warm relative to your baseline. And we acclimatize to different surroundings. So this means some people can find some things warm, some can find other things cold. typically we have warm fibers and cold fibers. Both of them respond between 23 and 40 degrees Celsius. It's important to see that if it gets too cold or too warm, warm and cold fibers stop firing respectively. So warm fibers typically fire between 25 and 45, but stop firing when it's way too hot, which is like 45 plus. And cold is 10 to 20 and stops firing when it's too cold, which is less than 10. And also we have a lot more cold fibers than warm fibers. So when skin is maintained at the ideal temperature, which is 30 to 36 degrees, you really sense thermal sensation. And that you can kind of understand that by our internal body temperature being like 37, so our skin will be slightly less than that, which will fall under this category unless we get really cold. Okay, so one more thing about thermoreception is that they have a tonic firing pattern. So tonic is the one that slightly or like slowly adjusts to its environment, so it continues its firing. As you can see on the diagram, we've got when it's at 34, there's a steady amount of firing, 36 increases, 38 increases, 42 increases even more, and then that's the warm fibers firing in response to increases in skin temperature. But you should also be able to notice that as time goes on, it sort of decreases in firing rate, since that's the adaptation happening. So it's clear to see in the cold fiber section at 24, you can sort of see that it fires a lot for like 4 seconds, and then sort of becomes a little bit more phasic. Well, it's still tonic, but it becomes more sparser. Proprioception are the third thing we're going to talk about. So proprioception is essentially the perception or awareness of the position and movement of your body. It's kind of called like your, I think it's like your seventh sense or something, because it is something that we're all, well, most of us have. And it's facilitated through two main things, which is muscle spindles and Golgi tendon organs. So muscle spindles are stretch receptors, Golgi tendon organs are tension receptors. And in terms of how they are organized within muscle fibers, so muscle spindles are in parallel with muscle fibers, Golgi tendon organ is in series, and we'll see exactly why in a short moment. So muscle spindles. Basically, I want you to realize that in a muscle, there's a bunch of different fibers, right? Our muscle spindle is kind of hidden within a particular type of fiber within our muscle. So this is a muscle that is not technically functional. It does stretch, but I mean, it does contract, but its main purpose is to give us proprioception, or like it's a stretch receptor. So these muscle spindles are inside what we call intrafusal fibers, which are specific fibers surrounded by group 1A nerve fibers, which detect stretching. So as you can see on the diagram, we kind of have our own muscle spindle covered by a fibrous capsule, which all muscle spindles kind of are, but our one is supplied by group 1A sensory neurons. We have extrafusal fibers as well. So our main two categories of muscle fibers is intrafusal and extrafusal. Intrafusal is within the muscle spindle. Extrafusal is not within the muscle spindle. And those ones are the normal fibers that contract, that we talk about all the time. And yeah. So muscle stretching, it essentially stretches the spindle. And this causes the activation or firing of the group 1A sensory neuron. Now, this concept is a little bit difficult to grasp immediately, but I'll hope that I'll explain it well enough. Basically, muscle contraction decreases the stretch, right? Since if you want to stretch it out, you got to stretch it out, but contraction brings the fibers closer together, so it decreases stretch, decreases firing. We have these things called gamma motor neurons. So typically, alpha motor neurons supply extrafusal muscle fibers, which are the normal ones, but gamma motor neurons supply intrafusal and contract the intrafusal fibers to match extrafusal and keeps the receptor taut, so that any consequent firing or stretching will cause firing. So what this means is, basically, if your muscle contracts and consequently your muscle spindle contracts, you kind of want your muscle spindle to relax when your muscle relaxes as well, because it means that your muscle has to contract beyond the initial point for it to fire again. Essentially, the spindle needs to adjust and return to the original firing rate. And it does this by gamma motor neurons, which basically cause the intrafusal fibers to match extrafusal fibers. But yeah, I hope that made sense. Ask any questions in chat if that wasn't clear. Second thing we're going to talk about is Golgi tendon organ. So this is for tension, muscle spindles to stretch. These are innervated by 1B, probably beta, I'm not sure, sensory axons, which synapse on the interneurons in the ventral horn. These interneurons may target the same alpha motor neurons of the same muscle. What this means is we can have a reflex arc. So if the we call the Golgi tendon organ reflex. So if the muscle is relaxed, once a certain amount of muscle tension is reached, and that's detected by the Golgi tendon organ, it can, through a reflex arc, cause muscles to be relaxed since it targets the same alpha motor neurons. So you can let go of a bag beyond a certain muscle tension, for example. A bit more straightforward. And that is the end of my part.
I'll get started on study design then. Yeah, hopefully my computer doesn't crash. Trying to screen share. Let's give it a shot. Always suspenseful. Okay. Doesn't seem terrible yet. Oh. Does seem terrible. I'm not sure if you can hear me. All right.
We can still hear you. We just can't see the screen yet.
Okay. Yeah, it seems like as per usual.
Uh-oh.
Yeah. I'll try and stop sharing.
Yeah, I think I should be able to stop yours like that. There we go. Perfect.
Oh no. Oh, he's still there.
Yeah.
Hey.
He hasn't left
This lesson, it happens every time, but I'm just a little bit optimistic. Would someone else be able to share study designs? Thank you.
Yeah, I can do it.
Perfect. All right.
Never fails to embarrass as well. All right.
Just give me like 2 seconds, I'll get it up.
Yeah, no worries.
Okay.
So yeah, we're going to be talking through study designs. This is really useful for your assignment that you'll have upcoming. I think it's what, ABM assignment, is that right? Yeah, so it'll be useful just to, I'm pretty sure most of your study designs will be sort of towards the end of what I'm talking about because they're relatively high quality. But it's worth getting a good overview of the different sorts of studies that we can have and how good they are, their advantages, disadvantages, things like that. So key ideas, we've just got some definitions here. A case generally is a person with the disease or the event of interest or the outcome of interest. A control is a person without the disease or event of interest. Sorry. Yeah, that's right. Exposure is the thing that is sort of precedes the outcome or that we suspect may have a link to the outcome. So it's something that affects the likelihood of a person developing a disease. Outcome, that's the outcome of the study. That's the sort of the result. So such as the development or the non-development of a disease. And then quantitative, you may be familiar with, it's involved with quantities, so numbers, measurable numerical data. And comparatively, then we've also got qualitative data or qualitative studies that we won't really discuss here because they're not super relevant in this context. And that's more descriptive data. So you can't measure it numerically. So for instance, how someone specifically feels a type of pain. Oh, it's colicky pain, it's sharp pain, it's a dull ache, that sort of stuff. Yeah, and in this presentation, we're going to go from lowest quality or lowest veracity to highest quality study designs. All right, so we've got this pyramid here. Top is highest quality, bottom is lowest quality. So meta-analyses are generally the highest quality. A meta-analysis will go into more detail at the very end because it's the highest quality, but they're sort of studies of studies. An RCT, or a randomized control trial, is of the experimental study design type. All of the other study types that we're discussing that are lower than that, for real. All of the other study types are what we'd call observational. So the investigator didn't assign the exposures. A randomized control trial, the investigator does assign the exposures, and we'll talk a bit more about that as we get into it. Next slide, please. Thanks, Jana. So yeah, here we've got sort of the breakdown of the main sort of quantitative study designs. So we've got that division between observational and experimental. So on experimental on the right, we can see that randomized control trial, which is the only experimental study design type that we're going to talk about today. I feel like I'm rapping a bit. And in experimental study design, we've got that the investigator did assign the exposures. So for instance, they will give controls just a sugar pill, and then the intervention arm of the trial, they will give the drug that they're testing. So they're able to choose. Now they want to make sure that they're randomizing it so that they're not biasing the data, but they actually choose that. Whereas an observational study design, you're looking at things that are going on and you're going, oh, some people are exposed to, say, some people are smoking and some people are not smoking. And you're going, oh, well, what effects does that have? Observational, you can't actually really prove causation. Obviously, the higher quality of observational studies, they can start to sort of strongly imply causation, but they definitely can't prove it. Whereas an experimental, generally, roughly, you can sort of prove some causation or some temporality, some this happened, then that happened because of this sort of relationship. Next slide, please. All right, so zooming into that observational arm, which is most of what we're going to be talking about. We've got a lot of study designs under that. They don't have a comparison group in the descriptive type, and they do have a comparison group in the analytical type. Observational studies are what I discussed earlier, but getting into the descriptive versus analytical. Descriptive, we're describing what's going on, and we're not really looking specifically at a comparison group. It's the lowest level of evidence because of that. And really, we mostly use it to generate hypotheses or to sort of observe rare occurrences, rare diseases, things like that. So it's useful for going, oh, this is information that we should investigate further. And for analyzing things where, say, there's only been like 10 cases of an extremely rare disease, it'd be very hard to sort of set up more general or wider studies around that. Analytical studies, we are assembling groups of individuals, and we're looking, for instance, if risk of disease is different for different individuals based on exposures, or if they were exposed or not to a certain factor of interest. All right. So we're finally on to our actual first study design, or study designs, really, I should say. So a case report, case study, and case series. So a case report is the same thing as a case study, that's synonymous. And that's basically a detailed report on the profile of a single patient. So you can see why that's relatively low in terms of evidence quality, because we're only talking about one isolated event. At the same time, though, very useful if there's a rare condition or something like that. I said usually, and then I didn't continue that text, but I'm okay with that. I think I got most of the key information down here. Well, comparatively, a case series is a collection of case reports, and it's sort of in order, perhaps. And they're looking at similar outcomes, similar disease. Okay, in terms of advantages, it's useful for the rare diseases or exposures, and it's useful for hypothesis generation. In terms of limitations, I mean, there are only two written here, but it's sort of because this selection bias is pretty substantial. And there's obviously a very strong risk of confounders. So other factors that are affecting it that we haven't picked up. Okay, on to ecological studies. So reminding you, this is one rank above case series, case reports. Okay, so we've got ecological study, that's also known as a correlation study. And basically we're looking at either different populations at one time or one population at different times. And we're comparing disease frequencies. So it's pretty fast, it's relatively easy to do, and it's quite cheap. And once again, not the highest quality, most rigorous research, but it's definitely useful for that hypothesis generation. But once again, we've got the risks of biases and confounding. It finds associations or correlations between an exposure, for instance, maybe. So this group, this specific population has a higher risk or a high frequency of this disease, sorry, and then this other population at this same time, or this population over different times, we can see their disease frequency is changing, and we can correlate that with, say, they're eating more mac and cheese at this specific phase. And so because of eating more mac and cheese, there's a higher risk of something. I don't know, I'm sort of spitballing here. You get the idea, hopefully. And also notably, we've got the ecological fallacy. So that's the idea that data from a population level doesn't necessarily apply to an individual. So while you might have some generalizable data, trying to then narrow that down to a specific individual may not actually be accurate. Yeah, I think in the lecture there's an example of Nobel Prize winners generally eating more chocolate. So there's a correlation, for instance, between those two things. I think because European countries tend to get more Nobel Prizes. I'm not too sure, maybe because it's closer to Sweden. But yeah, because of that, you might be able to come up with the probably incorrect hypothesis that eating chocolate makes you smarter or more likely to win a Nobel Prize. And that's generally, I think, able to be disproven. All right, sorry, we'll go on to the next one. Okay, so the next one we've got is a cross-sectional study or prevalence study. And this is the first one where I can actually give you a diagram. And you may notice as well, we've switched over from observational descriptive studies to now analytical. So stepping up the ranks here. So a cross-sectional study, that's taking a snapshot at one point in time. So we're measuring both the exposure and the outcome. And then we're measuring the presence of disease and the presence or absence of exposure. So you can see here we've gathered data on exposure and disease simultaneously. And then we've sorted our groups out into, have you been exposed or have you not been exposed? And then separating those further into, do you have the outcome of interest, the disease that we're looking into, or do you not? And so because of that, we can sort of estimate the prevalence of the disease of interest. It's relatively cheap and easy. And we're able to look at we're able to use it to sort of start to understand the cause or disease etiology. So the cause of disease, and once again, continue to generate those hypotheses for potential higher quality studies out into the distance. Nonetheless, it is difficult to make a causal inference. So you still can't really be sure that there's a causal relationship. It's still a correlation mostly. And because it's a snapshot, if there's something that's sort of flitting all around, what am I saying? If you have data that's sort of varying a lot over time, then a snapshot is probably not super representative of the overall view. So you may have different results. That's the limitation there. All right, getting into some more exciting stuff. Case control, and this is potentially confused with cohort studies, but I have a slide after both of those so we can talk through them. So a case control study oh goodness, death by PowerPoint, lots of words. Basically, we're looking at the outcome of interest, and then we are figuring out, okay, you have the disease or you don't have the disease. And then we ask them about, okay, what things have you been exposed to? Do you smoke? Do you drink? Have you been near a coal-fired power plant? I don't know, like things that you could hypothetically believe to maybe be an exposure that could cause the disease. And then basically by interviewing that, either by verbally interviewing patients or looking through historical records, you can once again start to recognize what might be risk factors involved in the development of disease. And once again, I think to some extent, sort of develop those hypotheses. Notably, it's characterized by a backward directionality. So you start with the outcome, you start with the thing that the result, and then you look backwards for the exposures. That's a very important concept there. And you can see down in this diagram under direction of inquiry about past exposures, it's pointing backwards. We have backward directionality. The data collection is once again at one point in time. So we're sort of doing this after the disease has already occurred, right? And we express the results in odds ratios, which I have a diagram of how to calculate it in like the space surrounding this slide, but I'm not sure whether that was copied over. Have a quick Google. It might make some sense. Otherwise, shoot us a message and I'll do a little learner on that. Okay, advantages. Once again, relatively easy, fast, and cheap versus its sort of often confused cousin, the cohort study. It's good for rare conditions and diseases with slow progression because we can choose to just study people with disease and making sure also that we're measuring controls as well to check that not everyone is eating chocolate, in which case chocolate probably isn't correlated with that disease. And once again, yeah, as I was saying, it's useful for identifying, oh, this might be a risk factor in the development of disease. Limitations we've got, it's really important that we carefully select, and I'm pretty sure relatively randomly select our controls. It needs to be representative of the population that we're taking the cases from as well, because otherwise we'd be biasing the sample. Other risks, we've got recall bias. So that's the idea that the person might not recall accurately the things that they were exposed to if you're interviewing people about it. Selection bias, so that's once again, that's selecting for a specific trait, intentionally or unintentionally, but thus skewing the data, and general measurement error stuff. And because we're using retrospective data, we actually can't establish incidence or prevalence. Yeah. Okie dokie. And now we've gotten to the cohort study. Exciting. So cohort studies, we've got, they can be divided into retrospective or prospective cohort studies. And I'll talk a little bit more about that on the next slide as well. But generally with a cohort study compared to a case control study, a cohort study, we look at the exposure of interest. So we've got that, we're looking at exposure first, not outcome. We look at outcome first in case control. We look at exposure first in cohort studies. So we go, okay, they've been exposed to this, or they've not been exposed to this. Let's follow these groups of people and identify of those who develops the disease and who doesn't develop the disease. Yeah, so a prospective cohort study, pro meaning for, so that's going forwards in time. Retrospective, retro meaning back, spective meaning like looking. So retrospective, we're looking backwards in time. Once again, I'll go into it a little bit more with the diagram in the next slide. We can put the results in either relative risk or odds ratio, but generally we get a relative risk for a cohort study. One of the best advantages of an observation of a cohort study is the fact that we're actually able to look at the temporality. So we're able to look at this happened, then that happened. Because we're following the cohort along a period of time and checking in with them intermittently, it means that we can actually see, oh, you've been exposed and then you've developed the disease, which is a very important idea. You don't want someone to have the disease and then be exposed, which you wouldn't be able to distinguish in basically all of the other ones, because we're taking them at sort of one point in time. We're taking a snapshot. So very important in establishing causality. Also, yeah, we are directly measuring incidence or when new cases are occurring as we're checking in. It's useful to carry out if an RCT, so a randomized control trial, which we'll talk about a bit later, is unethical. So if it's unethical to tell some people to smoke and some people to not smoke, and see if the people who smoke develop more lung cancer, that's unethical, quite obviously. So it's instead we can look at people who've chosen to smoke and people who've chosen not to smoke and observe their outcomes. And that way we're not instructing people to do things that we suspect may be quite detrimental to their health. Yeah, and it's also good for rare exposures and common outcomes. So we can also look at multiple outcomes. Because we're checking in with them multiple times, we can go, oh, have you developed this? Have you developed that? Have you developed this? We can check multiple things. And yeah, because of that rare exposure, yeah, it's a really useful study design. Limitations: expensive, time consuming, selection bias once again. So, you know, if we're not properly checking our controls, then that can result in skewing the data. I'll answer your question, Andrea, in a second. Loss to follow-up. So that's the idea that because we're checking in with people multiple times, if someone decides, oh, I don't want to participate in the study anymore, then we can't really force them to. And so that data, then we have to figure out how we'll deal with it. And that's sort of approached with intention to treat analysis, or another one that I'm forgetting. And also because of the long period to follow up, that can also result in sort of this drop off. But we really only want to be looking at common diseases or outcomes. We can't really be looking at rare diseases as much, because we're choosing to follow people generally before they've developed the disease. And so because of that, you can imagine that if we're only following 1,000 people and the rare disease only happens in 1 in a million people, there's a 1 in 1,000 chance that we pick up one case of that disease in our 1,000-person cohort study. Yeah. All right. So I see a question in the chat. Let me. Who's the control in a case control study? Is it someone with the exposure but not the disease? So in a case control study, we need to take a representative sample. If we just go back to the previous slide, Jen, we need to look at a representative sample of the population that we're also looking at the cases from. So say we're looking at, I don't know, the rate of breast cancer, then we'd want to be looking at a specific population, and we say we're looking in Victoria, and we're looking at women between the ages of I don't know, 15 and 70. Then we'd be basically making sure that we want to look at women between the ages of 15 and 70 in Victoria as well, for the controls. And we'd want to make sure that we aren't accidentally selecting more people of a certain ethnic group or of a specific age group. We want to make sure that we have a good spread of the people in that population, so that even though we're not investigating all of the women between 15 to 70, or the female Victorians between 15 to 70, we're still getting a good idea of what the general picture looks like of that population. All right. Also, some men develop breast cancer, so I probably shouldn't say just females. But all right, back to cohort studies. Actually, I think we're good to get to the disambiguation. Very colorful, fun, lots of colors, lots going on. All right, so case control versus retrospective cohort studies. So we've also got, I think, a prospective cohort diagram here, so that's useful. But generally, case control and retrospective cohort studies appear similar because we're both looking backwards in time. In a case control, we're looking backwards, but we're starting with, once again, very important to note, so we're starting with the outcome. We're looking at the thing that's been caused or that we suspect to have been caused. And then we're investigating exposures. Whereas in cohort studies, we start with the exposure. So cohort study, you start with the exposure. You start with the thing that you suspect to be causing disease. But we're looking at the exposure, and then we're investigating, well, what happens when they're exposed to these things? So for instance, I think in EBM assignment, we had the topic that I did was Hong Kong protests, and that related to different mental illnesses. And so they started studying, they recruited a lot of people into the study, and they were all in Hong Kong during the protests, and sort of they were investigating, well, how involved were they with the protests? What were their opinions towards the protests? And how did that affect their mental health? So development of, for instance, PTSD. So we're looking at the protests, the exposure. Were they experiencing the protests? Were they not experiencing the protests? And then from that, how involved were they? And then from that exposure, we're then looking, okay, well, what sort of mental health outcomes are they having? Things like that. Whereas a case control, you'd start with the disease, you'd go, oh, all these people are developing this disease. What have they been exposed to? And this diagram is from Wikipedia. It's really useful. I encourage you to look at it a bit, but I feel like I've talked through the main idea. All right, thank you. We've just got randomized control trials. Go through and systematic reviews, meta-analysis, and then we're done with me. Thank goodness. All right, so randomized control trial. This is our only experimental study design that we're talking about today. So that's where we have the person conducting the study actually has control over who's exposed and who's not. Okay, so those exposures are assigned to participants at random. So you sign people up to be in a randomized control trial, and ideally they don't know, and ideally you don't know who you're going to have actually taking the intervention or being exposed to the, and those who are going to be taking the placebo or being the controls. And the way that we refer to that is blinding. So the person doesn't know whether they're exposed or not. Single blinding is when the person who's actually receiving the intervention, so the trial participant, they don't know whether they've actually been exposed or they've not. And the obvious benefit of that is that they can't then be affected psychologically by knowing, oh, I've been taking the intervention drug. And so, wow, I feel a lot better, but it's actually just placebo effect. So it helps eliminate placebo effect. Double blinding, which is sort of like the main good one that we'll see. That's when the clinician, so the person actually like handing out the medications or administering them to the trial participant, they also don't know, am I administering the intervention, the exposure, or the control, the placebo, the non-exposure. And triple blinding is when the statistician who's looking at the data, when they're at least initially interpreting the data, obviously at some point they're going to have to know who was exposed, but they aren't aware. And so that once again removes that sort of bias of the statistician going through it. Advantages. RCT is a gold standard. Very good. Generally, they're free from unknown and known confounders. Because of that randomization, it means that we've got a really good mix of different factors. And so there's sort of this canceling out of them. It's unlikely that we're going to have some sort of big confounding variable, some big nebulous unknown factor that's impacting and skewing the data that we're finding. And that blinding reduces the placebo effect, as I said. In terms of limitations, as I said earlier, which is why we sometimes go to cohort studies, an exposure might be unethical to administer. You can't, like force people to go through things that you suspect may induce PTSD to see if that causes PTSD, that would be terrible. But what you can do in a cohort study is you can look at people who've been exposed and go, okay, well, let's follow you over time and see if you develop PTSD, so that you can help people. Ideally, yeah, so ethics very, very justifiably limits the sort of applicability or the applications of a randomized control trial. Very important. Also, some exposures, sorry, very briefly, some exposures aren't modifiable. So you can't actually like force someone to have a specific blood type or blood lipid pattern. Like you can't induce that. You can't modify it. You can't change it. So you can't randomly assign it. So you'd probably, once again, have to do some sort of cohort study or something like that, where you're following people along and seeing how they're affected. So you can't randomize. You can't do a trial, an interventional study on that. Okay, on to the final slide. Thank you, Jainam. So systematic reviews and meta-analyses, they basically are reviewing and combining the literature from previous trials on a specific topic. So effect of cinnamon on diabetes was one this year we looked at. And basically it looks at all of the studies that have studied, oh, does cinnamon have an effect on diabetes development or management? And then it comes to a conclusion based on all of the available good evidence. Generally, we're looking at combining lots of RCTs. So systematic review is really what we're sort of talking about there. A systematic review is when we're selecting all of those and we're combining them into looking at a specific question and finding an answer based on all of the available evidence. Cinnamon doesn't have a massive effect on diabetes management. And a lot of the trials aren't super high quality. For instance, a meta-analysis, that's really more to do with data management, and that but obviously like that meta-analysis is sort of really high quality because we're actually combining the data from a bunch of studies and using that to come up with a numerical finding, usually given within a systematic review. And generally, the big advantage here is that meta-analyses, they measure the effect size. So the averages that we take from a bunch of studies, that's going to be way more representative of the true effect and the effect size, so how strong the effect is, of, say, cinnamon on diabetes. Yeah, that's pretty much it. Thank you. Oh, and yeah, so just to sum up, we've gone through all the major study designs, and these are just some of those nice diagrams to give the holistic view. Thanks, and message any questions that you'd like.
Awesome, thank you to Kyle, and I'll be taking you guys through the next one, which is all of the exposure and stuff. So give me 1 second. All right, hopefully you guys can see that. So yes, long big name. This is basically elaborating on the stuff that we touched on earlier. So exposure to some sort of stimulus and how we actually draw a link causally between that and our outcome. So it's one thing to think that something's related. It's another thing to do a study, and it's another thing beyond that to look at your study and be able to say, yes, this actually proves that X is related to Y. Okay, so the exposure is quite nebulous, so it'll depend on the actual exposure itself. So if it's something which is fairly easy and objective to measure, then that's going to be evaluatable through things like blood tests, physical examinations, genetic markers, that sort of thing. If it's something more nebulous or which only the patient really knows, then we might need to rely on things like questionnaires. And obviously, in that case, we're going to be a little bit less confident in our data. That doesn't mean you can't analyze it, though, and we do go on through that. You just need to then acknowledge that that's a limitation. So in terms of what we care about with the exposure, we don't treat it as like a 1 or a 0. It's not you're either exposed or you're not exposed. We do need to actually care about the nature of the exposure. So we need to know the level of the exposure. So if we're talking about, say, radiation, we want to know how high the radiation was. And we want to know how long the radiation was, the duration as well. So we care about level and duration. And we also care about the way that the actual exposure affects the person. So do we see symptoms in this person immediately after they get exposed, or do we see symptoms only after a long period of accumulation? So yeah, they may not be interchangeable. So the damage caused, for example, by trauma, if someone punches you or something, that's not the same thing as someone pressing lightly against you for an hour, right? So you can't substitute the level for the duration. You do need to consider those separately in some cases. Okay, so dose effect and dose response. So the main distinction between these two is whether they happen to a population or just to an individual. So the dose effect says, okay, I've taken Bob over here and I'm going to give him increasingly high amounts of, let's say, radiation. And then that's tracking his response over time. The dose response is a little bit more tricky. So it's basically, I now have a room full of people. I've got Bob and all his friends. And we're going to subject them to increasingly high amounts of radiation. And then I'm tracking how many of them show signs of, say, radiation poisoning. This is a very, very unethical experiment. Please never do this, but that's the idea. Okay, so the dose response is tracking what proportion of the population has the condition. We expect that to eventually reach 100 at some point. The dose effect is just tracking the symptoms in a patient. So you can't do that as out of 100. That'll be depending on the actual symptoms that you see. And so confounding, Carl kind of like touched on this. I can't remember if he actually said the word explicitly, but it's the idea of when we have something that breaks the link or muddies the link between what we are trying to measure, so what we think is the cause, and the outcome that we're measuring. So in his example, there's chocolate. So we could say chocolate is a confounding variable to the link between something else. So the idea is confounding variable is usually linked to something else which also influences our main actual, like our exposure that we're tracking. So the thing that we actually cause. So we could say, for example, either being in Europe or being in a higher income country, both influences you to get more Nobel Prizes and also to eat more chocolate. And that doesn't mean that chocolate is linked to Nobel Prizes, because we can't, if we, for example, gave someone more chocolate, we would expect them to have more Nobel Prizes. If we took a bunch of people and gave them more chocolate, we wouldn't expect them to have, at some point, all got Nobel Prizes. And we can't draw that causal link. So that's muddying the waters, and we want to avoid confounding because it messes with our results and leads us to wrong conclusions, basically. Okay, so you might have seen some of this stuff, especially the targets are an absolute classic. You've probably seen these in high school at some point. So accurate is how close you are to the real answer, precise is how close your results are to one another. So I won't spend too much time on that because you guys probably all know that by now. We do have two more things. So reliability is basically if you write down your method, you give your results out, somebody comes across your paper and decide, I'm going to do this for myself. And then they do that, will they get similar results? If it's a well-designed experiment, ideally they should. Or they should get variance which is similar to your variance. If they're getting much higher variance, much lower variance, or say like completely different things, so they're getting a similar spread but at a different place, then that suggests that there's some error with the experimental design, which is preventing it from actually being a good test. Something else is affecting it, because your design should ideally account for pretty much everything that could affect the results. Validity is a really important one. So it's basically, is the test actually doing its job? And that's linked to what we talked about with confounding and control variables and whatnot. So the idea is, when we look at our results, can we be confident that they are driven by our independent variable? So internal validity is for literally the sample that you're studying. And so it's saying within these people, does this make sense? And then external validity is whether the results that we get from that test are actually generalizable to the overall population. So you can think about how a lot of different errors can lead to problems there. Okie dokie. So yeah, in terms of types of tests, this is, I think you potentially go over this in clinical skills as well. But we basically have two types of tests, heavy air quotes there, because the same test can be either of them based on the context. So screening tests are when the patient has no symptoms. We just give them the test because we want to, like, they might be in an at-risk population or something, and we want to catch that. Diagnostic tests are after the patient has shown symptoms. We investigate the symptoms using that. So screening tests generally are not used for, a test performed as a screening test is generally not used for diagnosis, but you can perform most tests as either. So you can give someone a mammogram if they've got no symptoms, or you could give them a mammogram after they've come in with a lump or something. So in the first situation, screening; second situation, diagnostic. And so in the first one, you might need to confirm that using other evidence. In the second one, since you've already got other evidence, you can probably take that through to a diagnosis. So it depends on the context there. Okay, in terms of errors, so we have two broad groups here. So the random errors are basically chance. There's something in the world which is affecting your results. So it could be natural biological variation. This is a big one with humans. We're not, abstract items. Each of us has lots of different quirks, and those will show up in your data as variants. And there's very little you can do about that. If you have a small sample size, then that increases the magnitude of those little variances. And that's why we care about a large sample size. And then finally, if you have imprecise equipment, then that can lead to high variance if it's not very good at accurately measuring, precisely measuring the data. So the reason I've starred small sample size there is because a lot of random error can be eliminated by using a sufficiently large sample size, because random errors will affect your precision, but not your accuracy, ideally. So they'll throw things off, but if you have enough of them, they should basically cancel each other out. So if I looked at the height of one person, then if that person was particularly tall, my estimate for the average height would be completely wrong. But if I had 20 people, there'll probably be some tall people there, there'll probably be some small people there, and there'll probably be some people in the middle as well. So if I look at a larger sample size, even though there's still random error within the data, it should be smoothed out more, and I should get a more accurate result. Then there's a systematic error. So these mean that there's an error with what you're doing, basically. So you're doing something wrong. So you've calibrated something wrong, and so you get a very close grouping, but it's not actually related to the real value. You can have selection bias. So this is something which affects external validity, so your ability to generalize to the wider population, because it basically means you picked a sample and the sample is not actually representative of that population. So like Kyle said, if you pick people and they're not actually a good representation of the wider group, then when you try to apply your findings from the small group to the wider group, you're going to have problems. And then recall bias. So this one is a specific one, often with case control studies, basically. So you ask a bunch of people, were you exposed to asbestos or something? And the people with mesothelioma, which you get from asbestos, are more likely to remember that they were, because they're looking for a reason. Whereas if you don't have an asbestos-related lung disease, you're less likely to care very much about when you're exposed to asbestos. And so you're less likely to remember it when a researcher asks you about it. And I think that's pretty much it, actually. Short and sweet. So if you guys have any questions, feel free to chuck them there.
So I think we got one question that was what specifically did you say about evidence and screening tests, etc.? I think that was the slide before the one we were just on.
Sure. I'll slide back up there. Yeah. So the basic idea is patient comes in, they don't have symptoms, but for whatever reason you want to give them the test anyway. So in the case of things like mammograms, we give them to women above a certain age, because they're in a risk group. So there's a decent chance they'll have something, even if we have no specific evidence for it. COVID tests, they're they can be done as either screening or diagnostic. So if they're just done as an abundance of caution, so like with athletes, they might test them even if they have no symptoms. So that's a screening test. If you have symptoms and you go to a COVID testing site and you get a test, then that's a diagnostic test, because if that comes back, you know that, if that comes back positive, you know that that's you have COVID, because you already had symptoms, and the symptoms have now been confirmed by the test. So again, you see a situation there where the same test, like the same procedure, can be used as a screening test or a diagnostic test. Is that kind of what you were going for? What you wanted to know?
Yep. Thanks, James.
Perfect. No worries. All right. I'll hand off.
Although notably, I'd say that a lot of tests are generally considered either screening or diagnostic in a clinical context.
Yeah.
You may have already said that, but yeah.
No, I didn't. That's a good point. So yeah, even though I said, like I said, you can use tests in both contexts, both ways, in some cases. In practice, that's not all tests. So like Carl said, a lot of tests are going to be mostly used as screening or diagnosis.
Hey guys, can you see my screen?
Yep.
So now we're going to talk about sampling. So when we perform study, it might be, or it could be very difficult to include the entire population in the study. So in these cases, we go for sampling, which we will take sample from this population and do the study for this sample. However, different samples will have slightly different results from the main population, and therefore sample mean will differ from the population mean. So to overcome this problem, we'll use confidence interval when making inferences. So confidence interval, we'll talk about it in the next slide, but it means that we are 95%, for example, confident that the population mean is within that range of the sample mean. Then we have another term called standard error, which is the average distance of the sample mean from the population mean. So basically, it means how accurate the mean of any given sample from that population when comparing to the actual population mean. So higher standard deviation means more variability, and which lead to a higher standard error. However, as the sample we take would be larger, so there will be less variability and therefore a lower standard error. We can calculate the standard error by this equation, the standard deviation divided by the square root of the population size. So yeah, as I said before, confidence interval means that, for example, 95% confidence interval means that we are 95% confident that the true population mean lies within this range. The confidence interval can be like 99%, 90 or 93, I think, percent as well. So for large samples, we can use this formula to calculate the confidence interval. However, in small samples, we need to use the D multipliers table that we can see here. Like in the exam, I don't think that you use these formulas. You just need to know them, because will not have like calculator in the exam. So data distribution. When distributing data, there's something called normal distribution, and we use this a lot in statistics and calculations, where this normal distribution means that 68% of the results are within one standard deviation, and 95% of results are within two standard deviation, technically 1.96. And therefore, the data can be skewed to the left, which means a negative normal distribution, or to the right, positive normal distribution. And we can determine this by the place of the mean compared to the median. So if the mean and median are exact same place, so this would be a symmetrical distribution. However, if the mean is on the left to the median, then we say that this normal distribution is negatively skewed, and if it was to the in the right median, it would be positively skewed. Then we have correlation and linear regression. Sorry. So correlation means if there is a relationship, a linear relationship between two variables, the way that we can interpret it, if the correlation is equals to 1, that means a perfect correlation between these two variables. If it's 0.3 positive or negative, that means there is a weak correlation between these two variables. However, if it's 0.7, that would mean there is a strong correlation between these two variables. However, correlation does not prove causation. Then we have a linear regression, when we try to make an equation to model the relationship between these two variables. And then we have multiple linear regression, which is the same as linear regression, but in these cases, we consider other variables. And so for each variable, there would be different equation. Now we'll move to the HLSD, human life development span.
Excuse me. We don't have the slides for that on the week 4 drive.
Which part is this?
This is the psychological medicine thing.
I think they should be there. I only uploaded them after I started the meeting, so maybe if you refresh, they should be there.
Oh, no wonder, because I downloaded it before the meeting started.
Oh yeah, that'll be it. Yeah. No, sorry about that. We did try to get them up all at once, but in this case, we had some tech issues, so that's why.
Yeah, that's all right.
Sorry, my bad. All right, so now move to the human lifespan development, and here we have some terms that we need to differentiate. So nature versus nurture, which is the degree to which genetic or hereditary influences or something we can't control, which is nature, and experiential or environmental influences, nurture, determine the kind of person we're talking about. Then we have continuity, discontinuity. Continuous means like height growth. It's a smooth progression. Like, yeah, for example, our height, there is a pattern we can see in there. However, in discontinuous, for example, there are like blocks or certain stages. Like there is no pattern, but there are stages or blocks. For example, When a baby, firstly they start crawling, they're crawling, and then they walk. Like there is stages. And we also have universal versus context-specific development. So universal, if it applies to everyone, and context-specific development, if it's like specific to individuals, or it's not universal or holistic. Yeah, and before I forget, many thanks to PSP Pinko, because they summarized this topic pretty well, so I used some of their summaries. And when we talk about human development, we also must talk about biopsychosocial framework, which are the factors or forces that affect this development of humans. So we have biological forces. It's all about genetic and health-related forces. So most which we can't control actually. Then we have psychological forces, which is about the personality or emotion traits or regulation. Then we have social cultural forces, which is about the environment and the culture around these people. Finally, we have life cycle forces, which reflect differences in how the same event affects people of different ages. For example, Alzheimer's diseases in a 50-year-old person versus 85-year-old person. So life cycle forces, it's about the individual differences or circumstances. Then we have these developmental theories with these fancy names. So I will not go through each theory because it would take a very long time, but you can look at it in your own time. So these theories basically, each theory has different stages that expect that these stages apply to each person or to each human when they develop. For example, you like see a lot of this Freud theory of development. It has 5 stages. However, Freud theory nowadays, we cannot imply it for all people. Actually, it cannot be implied for most of people. We have also Erikson developmental theory, which has more stages and it's a little bit more accurate than Freud theory. So each one of these theories have its advantages and disadvantages. Like there's no perfect theory because we are not all the same, we're different. So childhood development. First we have attachment. So attachment means that children seek attachment to other people to get the feeling of secure or closeness to other people. So according to Bowlby's evolutionary theory, children form attachment to adults as they are more likely to survive. So the children usually form these attachments with their parents because they are the closest people to them. However, they can form also attachments with any caring person around them. And in case of fathers, like children tend to spend more time with their fathers in playing. So children tend to seek out fathers for playtime. However, they tend to seek out mothers for comfort, and they form more attachments, much stronger attachments with their mothers, especially at their early ages. So we have these features of parent-baby interaction. We have firstly mutual gaze, which is about staring or eye-to-eye contact with children, parents with their children. And it was evident at birth that children prefer to look at faces more than other patterns and shapes. And it's more evident in females than females from infancy. Then we have dyadic synchrony, which is mutual attention and effective matching. We have conversations that's going with children and their parents, like children are very smart. They are smarter than we think. They actually can understand parts, or if it's not the entire conversation with them. And it's important for parents to talk to their babies from birth. Actually, so this study that suggests that babies talk to their babies or in front of their babies a lot tend to be much smarter than babies with quiet mothers or mothers that do not talk to their babies that much. Then we have social referencing, which is like babies are very sensitive. They can actually understand their parents' emotions. Then we have types of attachment. So we have an experiment, the strange situation experiment, where a mother and her child were put in the room, and then the mother leaves the room and stranger into the room, and the reaction of the children was recorded. And it was found that one type is secure attachment, which is found in 60% of children, where the child is comforted, crying stops, and they begin to explore. Another type, avoidant attachment, which happens to 20% of the children, where the child ignores or turns away. Then we have resistant attachment with 15% of these children, where they will be upset, remains upset when mother returns, and they are difficult to console. And finally, we have disorganized attachment with 5% of children, where child will be confused and unsure of reaction. And this is actually more worrisome, like this indicates that there is a problem. These children could be abused or any other problem. Then we have play. So first of all, make believe, like studies show that as the children grow, they form more relationships with other children. Like in the first years, they tend to play alone. However, when they 3 to 4 years, they can communicate with other children and play with other children. And as they grow, they form more playing relationships with other children. We have also gender differences. So between 2 to 3 years old, children prefer to play with peers of own gender. Girls tend to be supportive with other girls. However, boys tend to be competitive with each other. Then we have the parental influence, where like the role of parents when they engage their children with certain activities so that they will be more socially skillful. We have cognitive developmental theory. So we have this Piaget theory. Piaget is Swiss scientists which made this theory for the subcomponents of death. So we have pre-operational, where death is reversible, characterized by being still and eyes closed or departing. So children close their eyes or depart. Then we have concrete operational, which is meanings of universality, irreversibility caused by elements outside the body. Finally, formal operational, inevitable, universal, and casualty, cessation of bodily function. However, there are lots of disadvantages of this Piaget theory. For example, there's no consideration of environment or culture. Mistakes children make in pre-operational thought may be due to language development instead of cognitive developments and others. So as a result, another theories came up trying to overcome these problems with Piaget theory. Then we have attention. So it's like more likely just it's a memory thing. You need just to understand these terms. So attention is when sensory information receives additional cognitive processing. We have orienting response. So it's emotional and physical reaction to unfamiliar stimulus. And finally, habituation, sent reactions to stimulus after a repeated presentation. And you can see habituation in early stages of childhood development more likely. Then we have memory development during childhood. So during childhood, memory improves with age starting from birth. Newborns show habituation to stimuli, but memories last only a few seconds. And as they grow, they will have more durable memory. So their memory will develop until certain age, until they become very old, and then their memories will start again to collapse. Finally, we have adolescent development. So we have firstly puberty, which is dramatic increase in height, weight, and body fat distribution. So there are changes, physical changes occur with males and females. For example, secondary sexual characteristics appears with of pelvis in girls, in boys, facial hair, deepening in voice. And we have sexual maturation, menarche, onset of menstruation in girls, and we have also spermarche, which is the first spontaneous ejaculation of sperm containing fluid. And girls begin growth about 2 years earlier than boys. So girls with age 11 to 15 will become teenagers, and boys begin showing these secondary sexual characteristics between 13 to 17 years old. Brain development. So growth and metabolism requires more calories for teens, and boys need more calories than girls. So at the start of adolescence, the brain is nearly full-sized, and we have two systems, mature systems and non-mature systems. In mature system, we have the limbic system, where teenage are pleasure-seeking, and in non-mature, we have prefrontal cortex, where teenagers are, it's like self-control or decision-making. Then we have risk-taking, where increased sensation-seeking, and this sensation-seeking develop with females when they are 16 years old, so it's earlier than males, which develop this sensation when they are 19 years old. I think that's why females live longer than males. Then we have lifestyle, style, and behavior. So the development of teenagers are influenced by nutrition. As I said before, boys require more calories than girls. Also, we have they require iron to boost hemoglobin in boys for their muscle build, and for girls because they lost blood. And they also require much more calcium for bones. And here's a stat: most US teens consume enough calories, but most are poor in nutrition or they are too fatty. Then we have overweight and obese youth, which 30% of teenagers are overweight or having obesity problems. So there are factors that can influence this problem. We have genes, which they can't control, and the other one, energy imbalance, where there is imbalance between the input and output, or they're eating too many calories, but there is no enough physical activity. And successful interventions about setting goals about eating, exercise, and sedentary behavior only work with 20% of teenagers, and they are best to prevent than intervene. We have physical fitness. Teenagers are rarely get enough exercise, and boys do more exercises than girls. Then we have smoking, which leading to contributor to health problems. Nicotine is a known potent teratogen. And 2.5% of those 12 to 17 years old teenagers smoke every day. The average day of first cigarette for females is 15 years old. For males, 15 years old, and females 16 years old. So it's just information. I'm not sure if you have to memorize it. And for alcohol, we have this problem of being drinking, which is a major health concern for 18 to 19 years old teenagers. And no more than 4 standard drinks on single occasion reduces risk of alcohol-related injuries. Then we have sexual orientation, which I think we'll talk in much depth and details in the upcoming weeks. Not sure. So it's the patterns of emotional, romantic, and sexual attraction and sense of personal and social identified based on those interaction. And the sexual orientation is explored during adolescence. The males usually explore their sexual orientation before females. We have romantic relationship, which starts around adolescence. 50% of 15-year-old teenagers and 80% of 18-year-old teenagers had romantic relationships in the last 1.5 years. Early dating with many partners associated with problems in adolescence. And we have these gender differences in terms of attitude. So boys are more likely to describe first partner as casual date, and they have more positive feelings. And girls report stronger feelings of love for the first partner. However, they're more likely to experience feelings of guilt and fear. Sexual behavior is influenced by social environments, cultural environments. For years 10 to 12 students, 69% of them engaged in some form of sexual activity, 34% of them sexual intercourse, 59% they use condom as teenage, and as teens get old, they tend to decrease. This is because of ignorance, illusion of invulnerability, lack of motivation, or lack of access. And here statistics say that 11 of every 1,000 Australian teenage females who are 15 to 19 years old give birth, and this 4 times higher with Aboriginal and Torres Strait Islander. And finally, we have these threats to well-being. So main causes of death with teenagers are suicide, non-transport accidents, accidental poisoning, assault, and last we have event of undetermined intent. So accidental deaths often stem from high-risk behavior. And that's it for today. Thank you so much.
Thanks to Marwan, and thanks to all of you guys for coming along. Again, bit of a longer session, but even though despite the lack of anatomy this time, but yeah, we'll try and, as always, keep them as short as we can. Hopefully you guys got something out of it. Next week we'll be back to Monday, so see you guys at 9 AM.
Thank you.